these webinar series every Thursday, but also because I have the pleasure.
That's bad. Crap. Hold on. Sorry, Rui got muted.
Okay. No, I think
sorry about that.
I unmuted myself. No
All
problem.
Right, continue.
This
Sorry
just makes
about
it more
that, Rui.
fun.
Continue.
So, as I said, I was introducing Kathleen Daniels. Kathy works in our core facility at Memorial Sloan Kettering Cancer Center. Kathy, I can give you a brief introduction. Kathy had her bachelor's in bioscience back in 2011, I believe. She went to work to Regeneron at the Research Flow Core facility in 2013, where she very quickly went from a flow tech operator to a manager of the Flow Core facility. And we were very fortunate in 2018, in August 2018, to have Kathy join our Research Flow Core at Memorial Sloan Kettering, where she's been doing a phenomenal job in bringing the facility up to speed. I mean, it's a very intense, it's a big facility, but she's very driven and passionate, and not only she's been, as I said, able to really bring the Flow Lab up to speed, but she's also been involved in many things. She's a vice president of MetroFlow. In 2019, she was appointed as the ISAC SRL emerging leader, and she even has time to play around with parodies. And this is how fun she is in all her work and how passionate she is. So she's been writing parodies, writing songs and poems on flow cytometry and publishing them. And I encourage you guys to watch our YouTube channel for more of those kind of things. But this is just to give you an overview of how Kathy is. She, during this lockdown, she quickly put together a lot of ideas and pushed forward for virtual classes that, again, in collaboration with Flowtex, we've been putting out, and including some of these webinars that some of you have fortunately seen. And it's been, you know, this passion and drive that actually led Sloan Kettering to invite her to give a talk on this webinar series that we were supposed to have in April, about antibody validation. And they asked Kathy to give a presentation on antibody validation in flow cytometry. And so we took the opportunity to invite her and actually present this such an important aspect in flow cytometry, and to, you know, give us her view on best practices and antibody validation. And actually, I should say best practices, some of the things that she is very passionate about in flow cytometry. So before I give the floor to Kathy, I just want to remind everyone that if you have any questions during the talk, you can just use the chat function and just place your questions there. I will be moderating, and hopefully Joel from Baylor and Flowtech will be also looking out for these questions. And if we need to interrupt Kathy to clarify anything, we will during the webinar, or we'll just wait until the end and ask the questions for you, and you'll have those questions answered. So feel free to put those questions in the chat function, so that we can have a more interactive webinar. So Kathy, please, the floor is yours. And you may be muted. Are you muted? I can't hear you, at least.
I'm muted. Now I should be, right?
There we go.
Okay. Thank you. I'm assuming that was Joel or Paul, so thank you so much. Thank you very much, Rui, for the overly kind introduction. I very much appreciate it. And thank you to you, and also to Flowtex for being kind enough and inviting me to give this virtual classroom today while we're all working from home, or majority of us are working from home. This is a really exciting opportunity to be able to educate the community as a whole, and it's something I'm definitely very passionate about. So thank you, Rui, thank you to Flowtex, and with that, we'll get going. So again, as Rui described, today we're really going to be going over my views on antibody validation by flow cytometry, right? So what we're going to be discussing today throughout the duration of this classroom is we're going to have a discussion on what rigor and reproducibility is and why it's so important when we're talking about antibody validation and flow cytometry, right? And in order to get a better understanding of how we can carry this out, what we're going to do is we're going to take a brief look at how antibodies are really structured, different types of production methods, and why the validation of them is so critical, right? So why is it important to do? In order for us to be able to really understand that, what we need to do is we need to understand understand how to validate. We need to get a little bit of a deeper understanding of some methods that antibodies can bind to our cells that we're utilizing in flow cytometry. And once we know that, we can then move on to the different types of controls that are really necessary when we're validating. That's going to take into account positive controls, negative controls, what we can utilize for that. And then from there, you know, we can have an understanding of when we might have some issues with cross-reactivity, how do we test for that? And, you know, how can we validate our antibodies using all of these controls and understanding these principles? Okay. From there, we'll go over some common pitfalls that might come into play when we're validating our antibodies and utilizing antibodies in flow cytometry. And we're also going to be talking about best practices for optimal results, not only for antibody validation, but just for your flow cytometry experiments in general. And we'll wrap it up with some really nice available resources to you that, or any cytometrists that you can utilize to be able to find the best antibodies and move forward with your best experimentation possible, okay? So when talking about rigor and reproducibility, what we really need to understand is kind of what that is. So we want to make sure that we're always as rigorous as we possibly can be when we're designing our flow cytometry experiments, and that we have the ability to reproduce this, right? So the NIH describes it nicely on their website, where they describe that the two cornerstones in order for science to be able to move forward and advance is rigor in designing our experiments, right? We don't want any bias. We want to make sure we're utilizing the best practices, that we're really designing a robust experiment. And we need to make sure that when we design those experiments and we carry them out, that there's ability to reproduce that. And a really nice example of that, and the reason why it's such a important topic is, you can imagine us all as scientists, what we're trying to do is we're trying to build on scientific research and push forward. And the quicker we do that, the better, right? In the sense that we could really build upon the research of others in order to have our research grow and our knowledge grow. And back in about 2011, 2012, this kind of came to light in a very big way. And we can see here from this data where we're looking at reproducibility rates, what wound up happening is Amgen tried to reproduce some published data. I believe it was about 53 papers that they tried to reproduce in their own facilities to have a better understand or move forward with potential therapeutic options. And they were really only able to reproduce about 11% of what they had set out to do. And then Bayer had also done a similar study, and only 25% was reproducible. So this is something that can really set scientists back when we think about the amount of time and effort, resources, all of those things that we're putting into our research. If we're trying to reproduce and go based off of results from other facilities and we're unable to reproduce, that's going to set us back quite a bit. And there's a lot of different reasons that can happen. but we want to tackle that and make sure we're doing the best we can in order to prevent this reproducibility crisis from happening in our own facilities. Okay, so what we're going to be doing is we're going to be looking at flow cytometry and antibody validation, and most all people that are here will already have an understanding of flow cytometry and the technology in and of itself. But just as a brief overview, through this flow post-it that we have right here that the Sloan Kettering core facility, flow cytometry core facility puts out on every 2 weeks on Mondays, we come out with this educational content to be able to give people some tips and some best practices. And we introduced with what is flow cytometry when we rolled this program out in January 2020. And we can see here that, you know, flow cytometry, since it started being utilized over 50 years ago, is really an exciting tool that enables us to look at so much, right, from our single cell suspensions that we are prepping for this technology, right? So we can look at phosphorylation, we can look at RNA expression, apoptosis, necrosis, proliferation, so many different things. We can isolate populations via cell sorting or fluorescent activated cell sorting. And a common approach that is used is also immunophenotyping, right? The ability to pull out certain cell subsets based on staining with antibodies, right? And also utilizing antibodies that are conjugated to fluorochromes in order to pull out whether or not certain proteins are not expressed, or cytokines, so on and so forth, okay? So because we're going to be talking about antibody validation and flow cytometry today, it's important that we have a good understanding of what antibodies actually are, right? So most people will have a base level of understanding about this, but it's important that we recognize what the different parts of an antibody are. And this is just a schematic that's nice to show you the different parts of the antibody, because what's going to be critical to us as we take a look at this today is understanding the different portions and what they do, right? So you typically, this is an example of like a monomer of, say an IgG, immunoglobulin, and what we see here, if you're looking at the bottom portion, is you have this fragment crystallizable region, the Fc portion of the antibody, and you have the Fab region, or the fragment antigen binding region. And what we are going to be talking about today is both aspects of these antibodies and both parts of these antibodies. And what you'll notice is that in the Fab portion of the antibody, or the variable portion of the antibody, you have a paratope. And what that paratope is, it's a very specific region of the antibody that's going to hopefully recognize one specific epitope on a molecule of interest, on the antigen of interest, okay? The Fc receptors are pretty stable across isotypes and species, so that doesn't change, but the Fab portion is the variable portion of that antibody that we are able to utilize in order to be able to, for flow cytometry, to pull out certain populations, okay? So with that being said, all antibodies are created equal, or are they, right? So some people might say it's an antibody, that's all it is, it doesn't matter what type it is, but there really are quite a few differences in the methods of production of antibodies and how you can obtain them. So what I'm going to be talking about today is polyclonal, monoclonal, and recombinant antibodies. And when we're talking about polyclonal, what winds up happening is you immunize an animal, rabbit, donkey, goat, typically, with a specific molecule or antigen of interest, and then that animal then has an immunological response, antibodies are produced, and you wind up harvesting and purifying serum from that animal. And because the antigen that's introduced will have multiple epitopes on it, you're going to have antibodies that are generated against multiple epitopes. So each antibody that comes out, it's not just going to be one type that recognizes one specific epitope, but rather you're going to have a variety of binding capacities to different epitopes on there. Okay, so it's not specific to one epitope, and we're going to keep that in mind. Now, with monoclonal antibodies, with the advent of hybridomas, I believe in about the 1970s or so, some point around there, what wound up happening is mice were immunized, and they harvested the spleen and the B cells from those spleens and fused those with a myeloma, immortalized myeloma line to form hybridomas. So then typically what's happening is you have a B cell that's fused with a myeloma cell, and you get the hybridoma. And then through either serial dilution, or if you're utilizing a flow core or have a cell sorter, you can actually sort out single cells. And what winds up happening when you do that is you sort single cells typically into a plate, 96 well, 384 well plate, you grow them up, and then you can use a screening assay such as, ELISA-based assay with the supernatant to get an idea of the binding capabilities of specific secreted antibodies. And then you pick the best target based on that binding, and you go ahead and you utilize that antibody from that specific clonal population. And then you only have the antibody reacting to one specific epitope as opposed to the polyclonal where you have it reacting to multiple types. There's also recombinant antibodies that are becoming more and more popular, and they're available from a few different vendors. And what winds up happening is you kind of sequence that variable portion of the antibody, and then you introduce that via a transfection, I believe, into a mammalian cell line, and then you get recombinant antibodies that are produced that are very specific to the epitope due to that initial sequencing that was carried out. There are, like I said, different vendors that have this available. For example, there's even the reaffinate, or certain types of antibodies actually that have a modified Fc region, so that it's not the traditional type of Fc region of the antibody. I'm not going to get into too much detail about that today, but what I want you to understand is that when we're trying to have specificity in validating our antibodies, we want to make sure that we don't go ahead and use polyclonal and avoid them whenever possible, because they're going to bind to different targets, okay? So then, now that we understand the different methods of the different methods of production of antibody, we can have a better idea of why we want to validate, right? So one of the goals of validation is going to really have us ask the question of, does the measured signal from the cytometer have biological meaning? This is a really good way to put it, right? Because just because you see negativity or positivity on your cytometer when you're running your samples, does that have a true meaning to it? And really what we should see when we have a well-validated antibody in flow, the goal is that it should only react to one epitope that's specific to your molecule of interest, your antigen of interest, and that should be specific to your target species of interest, okay? So in order for us to validate that, what we want to do is we want to understand the different methods of antibody binding. I hope you're all okay with my very basic little cartoon here of a cell. It's not the most, I'm not a graphic designer, but I think it does its purpose. So when we're looking at this cell that is expressing a certain marker, right, which is going to be this little guy right here, you can have the fluorochrome conjugated antibody come in, and the antibody can bind to that epitope of interest on that antigen. And that's going to be an antibody antigen specific binding via the epitope of interest. So this is specific, and it's that paratope that I was mentioning earlier on the antibody, on the Fab portion, binding with the epitope on the molecule of interest. Alternatively, what you can have is you can have that Fc portion of the antibody binding to Fc receptors on the cells as well. This is also specific, and it's important to make sure we all understand that. Sometimes people think Fc-mediated binding is non-specific, but it is very much specific. And what the Fc receptors are, it's typically CD16, 32, 64. They're expressed on a variety of cells. And what can wind up happening is we have methods to be able to prevent this specific binding, and we're going to talk about that later. But when we see that, we want to prevent that as much as possible, because even though it is specific binding, it's not targeting that specific protein of interest for us or that specific epitope that we're looking for. Okay. Then lastly, what we can have happen is we can have an antibody conjugated to a fluorochrome that might go ahead and non-specifically bind. There are a couple of different reasons why this can happen, and we're going to be talking about that a little bit later. But these are the three different types of binding that I'm going to be talking about. So again, epitope, antigen, or epitope and antibody specific. Then we're going to have the Fc-mediated specific and non-specific binding. So when we talk about specificity, we really need to understand our target, and that's going to be critical when we're trying to determine specificity of our antibody. So it's important when we ask this question of, is our antibody specific, that we understand what it is that we're actually looking to go ahead and stain and or pull out in our flow cytometry experiments. A really good example of that is this example here, and I actually like to teach people from my my experience as much as possible, so that they don't repeat what I've done. And if I'm looking here at an experiment that I tried to run in the lab, what I did is I took unstained human PBMCs, and we had a very basic, like 6 or 7 color panel, just pulling out T cells, B cells, just very basic. To do something in our lab at Sloan Kettering. And what you see here on the left, if you can see my mouse, is that we have CD45 conjugated to Alexaflor 700 versus an irrelevant fluorochrome. Okay, so when I look at the unstained, and then when I look at the stained, what we see here, is we really don't see a shift up of that Alexa 700 signal. So if I'm looking at human PBMCs, and I stained with CD45, what I should expect, if I was correct in my assay, was for most all of everything to shift up, with the exception of red blood cells or anything that's contaminated in my sample that is not a leukocyte of interest, right? So I wasn't sure what happened. Maybe I didn't have the antibody. I kind of went through this whole process in my brain. Maybe something happened in the lab, an ARIA or the SH800, something like that broke down. I had to fix it. So what I did was I was like, oh, maybe I didn't add. I'm not really sure. But what I wound up doing at the end was I went back and I looked at my antibody vial, which is always the first thing that you should do. And I realized it was actually an anti-mouse CD45. So I didn't really go ahead and take a close look at that antibody, and I didn't have that time to really set aside and say, what is my target? What is my actual antibody that I'm looking at, and what's going to happen there? So this is a very blatantly obvious mistake that that I had made, where I used the wrong antibody, and you could tell that that antibody is not specific to that marker of interest. Okay, so we want to know our antibodies and what we're using, and that's going to be critical when we're having our discussion about specificity. Okay. So what do we really want to do? We want to be in control. Okay, any flow cytometrist should be a big fan of controls. And what we really want to ask ourselves is the question of, how can we know that the antibodies we have are specific to those proteins of interest, or or the cytokines, whatever it is that you're going to go ahead, surface markers, chemokine receptors. There's all these different things that we can take a look at. And what we need to do is we need to be able to validate our antibodies by testing them with appropriate cell lines or tissue. And when we're talking about that, we need to make sure that we have the appropriate positive and negative controls that we're going to be discussing a little bit more in depth, to make sure that we have the appropriate discussions about specificity and positive and negative controls, right? So when I'm talking about positive controls, the most important thing that we can do, or one of the things that we can do rather, is we can make sure that by utilizing cell lines that have been transfected to express a certain protein of interest, we can make sure that that antibody that we're trying to validate actually shows positive signal. So that's a good example of how you would have a good positive control to validate your antibody. So once you know that those cells really go ahead and express that marker of interest, you can validate the antibody. It's typically recommended to check the transfection, right, with another previously validated antibody that you have before you move forward with the new. Or what you can do if you're starting from scratch is you can use another method to validate that that protein is being expressed on the cells. Typically, you can do this with tagging, such as like a GFP tag or an HA tag that you come in with an anti-hemagglutinin antibody to validate that that protein is actually being expressed. But the important thing here is that we understand that we need to know with the cells or with the tissue that we're working with that the specific marker of interest that we're trying to have an antibody against is expressed so that we have a known positive signal that will come up. Okay. Then it's equally as important to talk about negative controls, right? Because a lack of staining is going to be just as important to us as a positive level or a positive signal that pops up when we stain. You can utilize knockout cell lines, right, where you're knocking out the specific gene that codes for that protein, and by doing that, you have a well-validated way of knowing that that protein is not being expressed. And then when you come in and you stain for that antibody, you shouldn't see a shift up of signal. Alternatively, if you're not working with lines that you can do a knockout and you're trying to work with primary tissue from human or something like that, you can go ahead and test the tissue of the same species that you know does not express that specific protein of interest and check for the non-specific binding or even cross-reactive cells or cross-reactive antibodies as well here. Okay, so there are methods that we can utilize via understanding our actual lines in tissue and also through technologies in the lab such as knockout cell lines that we can create our own negative controls. Okay, so when we're talking about the positive and negative controls, it's also important to remember cross-reactivity and what that's going to mean as we're validating our antibodies. So what we need to ask ourselves is whether or not the antibodies that we're trying to validate cross-react with different variants of the protein. If they do, what's going to wind up happening is our results won't be reliable and they won't be reproducible. And that goes back to our earlier discussion that we had about rigor and reproducibility, right? Because then if we use an antibody that we know might have cross-reactivity, we're not going to have other people in a lab somewhere across the world be able to, or even across the bench from us, be able to reproduce that data reliably. Okay? And a really nice example of that is here by a paper on cytometry part A, which I highly encourage everyone to visit, by Kalina et al. And it talks about antibody validation. And here in this specific schematic, they talk about, or they show the importance of testing for cross-reactivity. So in this example, what you'll see is they took a COS cell line. It's a fibroblast-like monkey cell line, and they introduced the human CD85 protein, or gene and protein. And they introduced different variants of that human CD85. And what we want to see when we actually test and try and validate our CD85D, or when they tried to validate their CD85D antibody, was they wanted to make sure that it was not cross-reactive across multiple variants, and that it was only reacting to the specific isoform of interest, which was CD85D, right? And what you can see is they tested two different clones. There was clone 42D1 up top in the top row, and then there was clone 287219 on the bottom row. And if you look, the red dashed line, is the unstained, and the blue line in the histogram is going to be the stained cells. Now, if you look at the top clone, 42D1, what you can see across the parental line is that we're not seeing any shift up in signal. And as we go across to the different variants that they had introduced of the human protein, you're not seeing any shift up of signal, which is just what we'd want, right? And then once you get to the human CD85D, you see a clear population that's pulling out of that protein of interest that they have the antibody against. When they went ahead and they tested a different clone, 287219, what they found was that as they were looking at the different variants, even though they know that it was not introduced to those cells, they found that the antibody of that specific clone was binding to the different variants. So that antibody is cross-reactive. That paratope that's produced with that clone has a level of binding to slightly different epitopes. And that's going to happen, and we need to understand the different isoforms that are available, that are going to be coming up with our different CD markers, right? And understand whether or not our antibody is going to have uniformity in recognizing those, or if it's specific to one isoform or one variant, okay?
Kathy,
Now,
just want to remind everyone that if they want to ask any questions, to feel free to put the questions in the chat.
Mm-hmm. Use that opportunity to take a sip of water too. Yes, absolutely, feel free. So then when we're talking about positive and negative controls and understanding how we can see if our antibodies of interest are actually going ahead and staining correctly, what we have to be mindful of is when we're trying to pull out markers that only pull out, or only we're really going to see with activation. So in this example here by Macker et al. In Cytometry Part A again, what we're looking for is, or what they were looking for was interleukin-2, right? So what we should understand is that the interleukin-2 is only going to really be expressed, or expected to be expressed, upon stimulation on our CD4 and CD8 positive cells, okay? And what we can see if we look down here on the bottom left is that we have an unstimulated control, and then up top you have a stimulated control, and that stimulation was done with staphylococcal enterotoxin B, or SEB. And this is a nice example and a really beautiful example of how we want to go ahead and utilize the cells that we understand from a biological perspective for negative and positive controls, right? So when we look at the unstimulated and we stain for that interleukin-2, what we see is we see a nice negative, because the antibody that we had used against IL-2 is not shifting up, okay? So it's not non-specifically binding. We're not seeing any background signal. So that's our nice negative control. And then when you look at the SEB stimulated sample, what we see is we see this population from the CD4 and the CD8 cells pulling out of the IL-2 positive in response to that stimulation. Okay, so this is a really nice example of how we can go ahead and be mindful of what the biology is in our experiment and use that to our advantage when we're validating antibodies. So that's something that might often come up if you're going to stain your cells and you don't really see a shift up in the signal. Understand whether or not that you should expect that signal. Okay, we also see here in this plot two other types of controls. I'm going FMO controls, just as a very brief explanation, even though it's not what this discussion is about today, is fluorescent -1 control. And what it is, it allows us to see the spreading that's introduced in multicolor experiments, so we can appropriately set gates. But what you can see here in this example is that the unstimulated was really the best control for the gating. And then lastly, you have the isotype control. We're going to touch on that a little bit later, but it really is not, and typically never is, the best control to use for gating, and we highly advise against it. Okay, and we're going to be talking about that and a little bit later. Okay, I think that's just some background noise. Okay. So as a review, when we're talking about activation, what we really want to understand.
Sorry, Kathy. Sorry to interrupt. Can you just go back, because somebody's asking here, how about the isotype control in this figure?
Yeah, so we're going to be, that's a great question, and we're going to be talking about isotype a little bit later and why it's not the appropriate control. but it's an antiquated method that's really utilized still in this day, and we try and educate people why it's not appropriate. I'm going to be touching about that in detail a little bit later, but this is a nice example that I can reference back to later as to why it's not the best control, because the isotype really is only going to tell us, you know, potentially if there is any Fc-mediated binding, or it might not even be relevant at all if that isotype might have some background or some low-level specificity of a marker that's on those cells that we're not aware of.
Right, and the other thing is, and this figure really raises that question, and somebody placed it here, is that you know, there's the question always of whether we should use the FMO control with our unstained cells or the stimulated cells.
Yeah, so I would suggest always being, you know, on the first run of your experiment, getting an understanding of what the expression of those markers are that you have, because maybe with unstimulated, you have a very, very base level of signal that's coming up. So you have to be understanding of your experiment and what type of controls are best for your experiment. So where you're going to have just CD4, CD8, or you know, certain markers that are expressed at a primary level, just yes or no, it might be a bit different as they have a continuum of expression or something like that. And if there's a base level of staining for unstimmed or something like that, I'm not an immunologist, we'd want to use the appropriate controls specific to our experiment. And you wouldn't necessarily always need an FMO for every marker that you're working with, and you have to understand what's going to be best for you.
Mm-hmm. Thank you.
No problem. So as a review of that, of activation, you know, certain markers really aren't expressed unless the cells are stimulated, and we want to use that to our advantage, like we saw with the interleukin-2. Another example of that would be CD69. We know that when we're looking at resting or activated T cells, there's going to be a clear difference between the CD69 expression, also on hematopoietic stem cells as well, for CD69 expression, and we can use this to our advantage to be able to test for positivity and negativity. Now, I mentioned earlier that we were going to talk about Fc-mediated binding and its importance. So when we're talking about Fc-mediated binding, we showed earlier through that little schematic that it is specific binding of the Fc portion of the antibody, that fragment crystallizable region, to a receptor that's present on certain cell types, CD16, CD32, CD64. And the reason why it's so important that we keep this in mind as we're trying to test our antibodies and validate our antibodies is that if you don't understand the cells you're working with, and if those Fc receptors are present, you're going to have false results, okay? This nice example here by Kuhn et al. Shows that when you have a blood sample that was stained for CCR5, chemokine receptor 5, you have 2.3 and 2.5% for expression for that population specifically. So it really wasn't a major flag that's kind of given off. But rather, when you look at tumor that you see here, what we're seeing is when we don't introduce the Fc block, you have an 83.1% population that's pulling out for the CCR5 CD11B. When you do an Fc block and you make sure that those receptors that cannot specifically bind to that Fc portion of the antibody are all kind of taken up, and you can't have that specific binding, what they saw with that tumor sample was that CCR5 CD11B population reduced dramatically and went down to 44.3. So that's a reduction of almost half, right? And when we're trying to understand biologically what's going on in our samples, if we can't be sure that the method of binding is antigen antibody specific via the epitope and the Fab region, we can't rely on our results. okay, it's going to throw everything off. And they showed another example of that here in tumor when looking at CD3 versus CD11B, where they had an 18% positive population, this double positive that was pulling out, that wasn't expected, that was completely gone once they introduced FC block. And I will say that this is something that's actually I've seen happen more than once in the lab and across different labs. And specifically, one example that inspired me to write a poem about it, that Rui was mentioning earlier, was one time I had a little bit of a later night sort with someone that had sacrificed quite a few mice, and it was a multicolor experiment. We ran all of our single colors and we did all of that, and everything looked good with compensation. And then when we went ahead and threw the actual samples on to sort, everything looked crazy, right? We had staining where we weren't expecting it, where we were not expecting it. We saw some kind of obscure populations, and we're trying to go back and see, like, did we run the controls correctly? Did something else go on with the instrument? Like, what's really happening here? And then the kind of light had gone off in the researchers' head, and they realized that they forgot to add FC block. So make sure that you're utilizing that and understanding the cells that you're working with and whether or not the FC receptors are present. And there are different methods that you can utilize in order to block the FC receptors. And while I'm not going to get into all the different types, whether you're using commercial block or you're using purified IgG or whole serum, know that there are resources available to you to read into this in the literature. Specifically here, there was an in-depth study done by Anderson et al. In cytometry part A again. And I'm going to keep referencing back to cytometry part A, as you can see, because it's such an important resource to us. But you can utilize this to your advantage to look, but always make sure that you understand methods of binding.
Right. So a few questions here of course, on FC blocking. It's a typical, debatable question. One of the Sarah Schneiders online is asking, is FC binding still a problem when the species of the antibody and target are different?
When the species of the antibody and the target are different. So you have, what you'll wind up having is you'll have your antibody that's raised of a specific, or if it's a specific subtype, you'll have mouse anti-human antibody, and that can go ahead and it can bind to the FC portion of the receptors on the cells, to my knowledge. And you want to make sure that you're going ahead and blocking appropriately with the specific subtype, or with the specific blocking reagents. It's my understanding.
Right, and actually somebody's asking here, and this is also the theme of one of the papers in the late 90s about isotype controls. They're asking here, is Fc-mediated binding the reason that we need isotype controls?
So that's something that people might utilize isotype controls for, but when we're talking about isotype controls, and we're going to bring it up a little bit later again, we have to be very, very careful about the way that isotype is actually raised. It has to be raised under the same exact conditions of our same antibody. It has to have the same fluorescence to protein ratio. It has to have the same reagents that were utilized. So there are so many different variables when we're talking about comparing an isotype to our antibody of interest, because it's generated in a different way typically. And we want to be very mindful of that when we're trying to look at them. So what I would highly suggest is use the correct biological controls with either knockout or transfection, where you're introducing the protein, so that you can make sure that what you're looking at is true signal that's expected by the binding portion of the antibody.
You may not be, I know you're not an expert in this field, so you may not be able to answer this question in particular. But if anyone here online can answer in the chat, that would be nice also. But I'll ask you anyway, Kathy. This question is, in your opinion, do you think FC blocking should be a standard of practice for MRD analysis in leukemia? Like for instance, in AML MRD or MRD?
So for that one, I will absolutely defer. I'm way more of a flow cytometrist than I am, an immunologist. I'm definitely learning, but I would have to defer on that question because I don't want to do it the injustice by
I think I think there are some flow experts here, clinical flow experts, so maybe they can chime in.
Yeah, and what we can always do with some of those questions that might be a little bit more specific from a biological perspective, we could always, if it's not answered in the chat, I'd be more than happy to take those questions and take the contact information to reach back out to people after the talk, after I've, after we've reached out to the appropriate parties for that.
Okay, thank you very much.
Thank you. Okay, so kind of jumped straight from blocking reagents. So we just spoke of two different methods of specific binding, all right? So we're talking about the epitope and the Fab portion, and then we also spoke of the Fc specific binding. Now we're going to be moving on to some different ways that we want to make sure that we're not seeing non-specific binding. And the first recommendation that we always have for that is to make sure that antibodies are titrated, right? And the reason why that's so important that you'll see as we have this discussion is that what can wind up happening is if we add too much and it's in excess, you can actually see some non-specific signal, even though and a combination of specific and non-specific binding. And we want to rule out and prevent that non-specific binding as much as possible. So once we're confident about the specificity of the antibody, then the titration of the antibody is critical to ensure we're using it at the correct concentration. And what you can see here on the left is a concatenated file where we have concentration 0 up to 5 micrograms per mil of an irrelevant, it doesn't matter to us what marker we're looking at, so it's just antigen F, BUV496. And what winds up happening is as we're increasing the concentration of our antibody, what we see is that positive population is shifting up, 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 and then it plateaus, right? And if we're looking at the negative, we see that negative, as we're oversaturating or adding too much antibody, starts to shift up due to non-specific binding. So oftentimes, the antibody vials will come with a recommended volume per test from vendors that don't tell us exactly what the concentration is, and it doesn't take into account the tissue type you're working with, because there's going to be different levels of receptors on different tissue types under different conditions. So you really need to titrate the antibody of interest on your cell types of interest so that you're making sure that you're using the correct concentrations, so that we don't see non-specific binding, and also so that we don't waste reagents, because typically the recommended volume per test and or use per test is a little bit too much, or maybe sometimes a lot too much. So typically our recommendation is to start off at 10 micrograms per mil. This example shows 5, do 8 serial fold dilutions down. And when we're determining the appropriate concentration to use, what we really want to do is we want to look at the separation index or the stain index. There are different methods, but we want to utilize a method that takes into account the positive and the negative, taking into account that spread of the negative as well, so we can get the best separation between our expressing cells and our non-expressing cells. So what you can see here is we calculated, the separation index, and when we did that, you can see this is the highest separation that we're seeing right here, and that was actually the 1.25 micrograms per mil in this example. Now, I'm going to do a little plug again for our Flow Post-it program, because we have this program that we come out and we generate these informational slides. Our most recent one that was put out by Joanna in our facility just this past Monday was all about antibody titration for flow, some useful tips, why it's so important, and what you can do to calculate the appropriate concentration of antibody. Now, that being said, the conversation can go a little bit deeper about that, because you don't always need to use the saturating level of antibody. And that can be a deeper discussion about multicolor panel design and best practices there. And we could always discuss that later. But when we're talking strictly from a perspective of validating and making sure that we're seeing specific binding, that's when we want to make sure that we're titrating and using the appropriate concentration. Okay. So going back to that example I put in before, where I incorrectly had the wrong antibody, I want to show you an example of why more is not necessarily better when we're talking about antibody. So this was going back to that same human PBMC sample, CD45. Before I went back and I looked deeper at that vial, what I did is something that is definitely not the best practice. I was in, and I want people to learn from my experiences of why this is such a bad practice. Because what I did was I went back and I said, oh, I just didn't add the antibody. I took a couple more microliters, I threw it in, I let it incubate, and then I threw it back on the cytometer without even washing. Okay, and what wound up happening is I did get that shift up that I was expecting of everything showing positive for CD45. So what wound up happening is I just added, I did not wash sufficiently, and when I did that, I did get a shift up, all from non-specific, and this is not typically normal level of non-specific that you would see, but it's a reason why, we want to make sure that we're using the appropriate concentration, that we're making sure we understand what we're looking at. And I very clearly had a false positive due to non-specific because of multiple bad practices. Okay, so don't just add more antibody. It's not the correct, it's not the correct practice. Okay, the next method, so I'm just gonna take a sip of water. Not used to talking this much nowadays. The next method of non-specific binding is going to be a quick discussion about how certain tandem fluorochromes can bind to certain cell types, specifically monocytes and macrophages. So this is a good example here from BioLegend, where they show that fluorochromes can actually have an interaction with the specific cell types. And what you see here is with CD3, CD14 example, and then CD56, CD14, when we have specific tandems such as cyanines or, even some of the fire dyes and dazzles, you might have a shift up in signal where we wouldn't expect because it's the fluorochrome binding to certain cells. So they use their specific commercial reagent to prevent that from happening. And it's also available from different vendors, not just BioLegend. This is just a nice example from them. And there's also, I believe, some other non-commercial available reagents that you can utilize for this as well. But we want to make sure that the staining that we're seeing is specific and not non-specific due to this fluorochrome interaction with our cells. Okay. Another way we can go ahead and make sure that we're not getting any fluorochrome-mediated binding is through the use of isoclonic controls. It's not something that's often discussed as much, but what winds up happening is you have an excess of cold antibody that doesn't have the fluorochrome conjugated to it. And when you have that, what winds up happening, because you have that excess, you have is that unconjugated antibody that comes in the same little silly schematic, and it binds to those antigens of interest. And when you come in with your fluorochrome conjugated antibody, if you don't have any, and it's a little bit of a quick animation, but if you don't have any fluorochrome mediated binding specifically, you're going to have no shift up in signal. Alternatively, if you come in and you do that same isoclonic control and you have a smaller amount of antibody that has the fluorochrome conjugated to it, and you do see a shift up in signal on the cytometer, that's due to fluorochrome mediated binding. So that's how you can rule out that the antibody might be interacting non-specifically via the fluorochromes with the cells through the use of an isoclonic control. And something like this, where we see those antibodies binding and we see the shift up in signal, is what we wouldn't want to see. Okay. Then, as promised, we're going to talk about isotype controls and why we don't want to use those. It's not something that we want to use for validation. It's not something that we want to use for defining positivity. It's really an antiquated method, like I mentioned earlier, and the determination of positivity should be done with the appropriate controls, not isotypes, right? So we want to use either unstained, FMOs, unstimulated, mock transfected. There are all these different controls that we can utilize to make sure that we're gating correctly and making sure that we're understanding positivity correctly. And like I said, I mentioned before about how the isotype was raised and how it would have to be in the exact same conditions, and how it's very hard to control for all of that. And while that's not the topic of discussion today, you could always reference the cytometry Purdue list. For example, here, this is about in 2005, I think, where Mario Roeder goes into a deeper discussion on why it's so, what you'd have to take into consideration when talking about isotype controls. So I think the subject on that one was isotypes intelligent design for science. So please, I encourage you to check that out if you want a little bit of a deeper explanation. Okay, so when we're talking about when we need to validate our antibodies and why we have to do it, when do we have to check our new antibodies if we're looking at a similar marker or that we've already had a validated antibody for? What we really need to do is we need to test if there's a new lot. We need to test if there's a new fluorochrome conjugation that we're using, vendor, clone, all of these different things we'll need to test for. I can say from my own personal experience, I've actually done a test of a CD4 kit all conjugated to different fluorochromes, and what we see in that, what I saw when I was doing that antibody titration of the CD4SK3 was that all the different fluorochrome conjugated antibodies had different optimal concentrations, right? So there was a broad range of optimal concentrations throughout the different fluorochromes. So you can't just switch to, if you're using the same vendor, same antibody, same clone, you can't just go ahead and assume it's going to be under the same conditions and that it will work just fine. I've also seen antibodies, that maybe it's just a new lot that gets sent out. Everyone assumes it works. They go ahead and they use it and there's no signal. So I encourage you to validate when you get new lots, and then also make sure that if you have something that you know is not expected, where you're not seeing a positive signal or you're not seeing a negative signal that you know should be there, reach out to your vendor reps, really talk to them, because these antibody vials are not cheap. And from my experiences from a variety of vendors, they'll really work with you to understand what was happening and make sure that the correct, and they reach the correct mediation. Okay. So when we're talking about all this, we also need to consider and validating antibodies. We need to have our discussion about best practices that we want to utilize. And most important, one of the most important things that we want to discuss is how once an antibody has been validated for other applications, that's not a green light to go ahead and use it for other applications. So for example, Western blotting, IHC, if you have antibodies that are validated to work for those specific technologies, you cannot just simply transfer it over for flow cytometry and expect it to work, right? There's a couple of different reasons for that that we can always discuss if everyone would like, without getting into too much detail, but we have to understand that different technologies, the proteins themselves might be in a different conformation, and the method of the binding of the antibody can be impacted there. Okay. Then also very important is we want to be careful about how we dissociate our adherent lines or our solid tissue. Being mindful that as you're doing any enzymatic digestion, you can actually alter those epitopes that you're trying to go ahead and stain for. So if you're coming in with, trypsin, acutase, cell dissociation buffer, there's all these different methods you can use for enzymatic dissociation. What you want to be mindful of is that if you're being too harsh and the reagent is too harsh or whatever it is, you can actually alter that epitope and it can actually eat away at a little bit. And then once that epitope is altered, the antibody that's coming in, even though it might be very specific to a certain region on that molecule of interest, if it's altered, it's not going to bind. So I would suggest troubleshooting your dissociation reagents and making sure that they're appropriate for your considerations and for your experiments. Okay, very briefly, just avoid secondary antibodies whenever possible. From my experience, I can say anecdotally they've been pretty sticky, not the best. So that's if, what people utilize, maybe if they can't find a directly conjugated fluorochrome, antibody fluorochrome conjugate, they would come in and they would, say, for example, use a mouse anti-human and then antibody if there was no direct conjugate to that, and then come in with like donkey anti-mouse or something like that, secondary, that is then conjugated to a fluorochrome. They're not the best, so what I would always suggest is that people go ahead and try and do their own in-house conjugation, or if they're trying to work with high quantities, you can always get custom conjugates, even though it might be a little bit more pricey. Okay. Then also very, very important, we talk about this all the time, is that antibodies can actually non-specifically bind to dead cells. So the inclusion of a viability dye is going to be incredibly critical if you want to make sure that you're getting the correct statistics, right? If you're getting the correct representation of positive and negative, because you can very, very easily have a population of positives that are showing up. But once you add in a viability dye, that's going to disappear. And just because you're fixing your cells doesn't mean you can't use a viability dye. There are commercially available amine reactive dyes that you can utilize that are amenable to fixation, or you can also use the DNA binding dyes that are available as well if you're using a live prep. Okay, lastly, a nice available resource that I just want to discuss very briefly through a video is actually BenchDye. So this is a nice tool. It's not an advertisement. I don't work for them. It's just a nice example of a tool for finding antibodies. So it's free to academia, and what can wind up happening, I'm just gonna actually go back a little bit, sorry about that, is you can go to this website and you can go ahead and start to explore the antibody that you're looking to find. And if I come here, for example, and I started looking for CD4 on this website, that's available to us, I can then go ahead and start looking for the application specifically that I'm trying to utilize, so flow cytometry. I can go ahead and look for what type of species I want it to react with, so CD4 human. I mentioned before, I use that the SK3 and clone, so I can come and look at the clone ID, it's SK3, and then I can look at conjugation. There's all these different options. So if I want a specific fluorochrome conjugate, I can come and try and define all of these options. And then it's going to give me times when this was published. When an antibody that they found with all these, that met all these criteria, was actually published in literature. This is a nice example here, because what you saw, I'm just gonna go back a second, is that that was actually referenced in an OMIP, which is an optimized multicolor immunophenotyping panel that's published through Cytometry Part A. Okay, I'm just going to reference that a little bit later as well. But you can utilize this to be able to explore antibodies that are of your specific clone of interest that have already been published. So you can then hopefully have a good idea of a better starting point. So it's a nice resource that's available. They also have machine learning, prediction of potential cross-reactivity. I'm not going to really go into that, but you can feel free to go ahead and look at BenchDye and explore the tool that's available to you there. Okay, and it'll also take you to the vendor site and all of that. So in summary, I think we're getting a little bit close, and just a couple minutes over. What we want to do is we want to make sure that whenever possible, we utilize monoclonal or recombinant antibodies, and we steer clear from the polyclonal so that we don't have multiple epitopes that are going to be recognized. We want to make sure we understand our target from a biological perspective and determine the appropriate protein variant for our purposes, validate our antibodies with the appropriate positive and negative controls, and really make sure that it's antibody antigen driven. And that goes into the Fc blocking and making sure that we're preventing that Fc-mediated binding. And we want to make sure that we prevent any non-specific binding as well through the titration of our antibodies and understanding if there are any fluorochrome and cellular interactions. And always utilizing the appropriate controls after we validated our antibodies to make sure that we're getting the correct gating and all of that. Okay. So with that, here are some more available resources to you. The FCCF, or the Flow Cytometry Core Facility at Memorial Sloan Kettering, has our own website, fccf.mskcc.org. And we also have our Twitter at FlowMSKCC is our handle. There are some regional user groups that have in-person, and in our current climate, a lot of virtual available educational opportunities, specifically FlowTechs, we're having this webinar with, or classroom with today, and also MetroFlow, which is our regional New York, New Jersey flow cytometry users group. And then we also have Cytometry Part A, and you'll have seen that we had quite a few resources quite a few citations from there today. And specifically, the first link is about antibody validation in flow cytometry that was just published back in 2019 that will go into a whole lot more detail on everything I spoke about today. And I highly encourage everyone to check that out. In addition to the optimized multicolor immunofluorescence panels that you can use as a starting point when you're trying to develop your panels and get good antibody clones and everything like that. Okay. So with that, I want to say thank you. First I want to thank the Flow Cytometry Core Facility Group at Memorial Sloan Kettering Cancer Center and all of the members of our group who've been phenomenal throughout all of this before, during, and they will be after. And specifically, I really wanted to say thank you to Rui, who's been an unending, never-ending support, and he's been amazing throughout all of this and went over the slides with me, in a lot of detail. In addition to FlowTex, and they've not only allotted me this opportunity, but were very supportive in the content as well. So we thank them for being so wonderful about providing this educational content to cytometrists kind of worldwide. And then lastly, I would be remiss to not thank Pat Burfind, who is my original mentor in flow cytometry when I started at Regeneron Pharmaceuticals back in 2013. She's since retired, which is sad for the community, but she was a wonderful resource. And with that, I say thank you and open it up to any questions that I will hopefully be able to address.
Thank you very much, Kathy. This was very, very useful. I'm trying to see if there are any more questions here. We've been addressing some of the questions on isotype controls and FC block throughout the presentation. So I'll just wait a few moments to see if any other questions are coming through.
And what I didn't mention, I should have actually put this in the slides, is my email is danielk1@mskcc.org if anyone has any questions, but I believe this is being recorded and we'll have this available for everyone before and after.
Correct. This is being recorded and we'll put it up on the FlowTex YouTube site.
Yes. Yeah. Thank you, Joel.
Well, I guess that's it. So if there aren't any other questions, Joel, do you want to wrap up?
Yeah, well, sure. Yeah, well, thank you so much, Kathy, for this wonderful talk. I appreciate it. If we end up, we'll go through the chat log, and if we've ended up missing anything, we'll send it on through to you and then send it out. Otherwise, we will be posting this on our FlowTex YouTube page, and we will be sending out a link to that as well to hopefully to all the registrants for anyone who happen not be able to make it. So if there's any questions at all, you can always email me at cederstra@bcm.edu. Most people have my email address because they hear incessantly from me. So once again, thank you so much, Kathy.
Thank you so much, everyone, and stay safe.
Okay.